I want you to picture a scene. It's a quiet Friday afternoon in the middle of winter.
Usually 1953, right?
Exactly. January 30th, 1953. And we are inside this drafty office at King's College London. A research scientist named Maurice Wilkins is standing by a metal filing cabinet.
Right.
He slides the drawer open, the wheels grinding softly against the tracks, and he pulls out a photograph. He turns around and he shows this photograph to a visiting researcher who's just come down from Cambridge, a young American named James Watson.
But, I mean, it's important to clarify that it is not a standard photograph. It's an X-ray diffraction image.
Right, yeah.
It's basically
Yeah.
a small square piece of film with a very specific arrangement of dark spots on it.
And Watson looks at this film, and according to his own later accounts, the moment he sees it, his mouth falls open. His pulse literally begins to race, because this isn't just any image pulled from a drawer.
No, absolutely not. It is an exceptionally sharp, incredibly hard-won X-ray image of a single DNA fiber. It is so clear and so mathematically precise that the basic geometric shape of the DNA molecule, I mean, the secret to biological life itself, it can be read almost directly off the film if you know what you were looking at.
But the most crucial detail of this entire scene is who is not in the room.
Right, exactly.
Because the person who meticulously took that photograph, the scientist who spent months, literally months, perfectly calibrating the equipment to bring that invisible structure into the light, she's just down the hall.
Her name is Rosalind Franklin.
Rosalind Franklin. And she has absolutely no idea that this filing cabinet has been opened. She has no idea her data is being shared with rival researchers. And, I mean, she certainly wasn't asked for her permission.
She is just sitting in her own workspace, methodically working through complex mathematical calculations based on that very photograph.
Wow.
She fully expects to publish her conclusions on her own timeline. She's completely unaware that the ground has just shifted right beneath her feet.
So, you've probably heard of Watson and Crick. If you've taken like a high school biology class, those names are burned into your brain as the discoverers of the double helix.
Sure.
But today, our mission for this deep dive is to unpack one of the most famous, most mythologized moments in the history of science. We aren't just doing the standard victory lap here.
No, we really have to look at the mechanics of an unauthorized data transfer that actually made that victory possible in the first place.
Exactly. We are looking at the brilliant experimental physicist who was kept entirely in the dark and a legacy permanently complicated by tragedy. I mean, you really cannot understand one of biology's biggest breakthroughs without understanding Rosalind Franklin.
You really can't.
Yeah.
And the standard narrative usually treats this discovery as a story of spontaneous theoretical genius.
Right. Wait, like two guys in a room?
Yeah. Exactly. Two brilliant minds playing with cardboard cutouts and wire models until they miraculously cracked the code. But the reality is a story of documented transfers of raw data, incredibly difficult working environments, and the hard, unglamorous labor of experimental physics that always underpins these theoretical breakthroughs.
Yeah.
To really grasp the weight of what happens in that office in 1953, we have to start much earlier. We have to understand the specific, formidable skill set Rosalind Franklin possessed before she ever set foot in King's College.
Because there is this terrible tendency in popular history to frame her as just a junior lab assistant. Like she just got lucky with a camera one afternoon.
Yeah, and that is a fundamental mischaracterization of her standing in the scientific community. I mean, Rosalind Franklin was born in 1920 in London into a prominent, politically active British family.
Okay.
And from the very beginning of her academic career, she demonstrated this staggering, rigorous intellect. She studied physical chemistry at Cambridge, focusing specifically on physical chemistry and crystallography.
I want to pause on that word, actually, crystallography, because this is kind of the bedrock of everything she achieves. If we don't understand what the science actually entails, her work just sounds like, taking pictures.
Right, just snapping photos.
Right, and I've seen these X-ray diffraction photos. They just look like blurry smudges and dots on a gray background. So, how on earth do you get a three-dimensional shape from a smudge?
Well, think about it this way. Imagine trying to guess the shape of an ornate crystal chandelier hanging in a pitch-black room.
Okay, I'm with you.
You aren't allowed to turn on the lights, and you are allowed to touch it. All you have is a flashlight. You shine the flashlight at the chandelier, and you look at the shadows and reflections scattered on the walls around you.
Like bouncing off the glass.
Exactly. Based solely on the angles and intensities of those scattered patches of light on the wall, You have to reverse engineer the exact placement of every single piece of glass in that chandelier.
Oh, wow. So the flashlight is the X-ray beam, the chandelier is the microscopic molecule, and the wall is the photographic film.
That is the essence of X-ray crystallography, yes. You fire a beam of X-rays at a crystallized substance. The X-rays hit the atoms inside the crystal and bounce off. They diffract.
Right.
And when those diffracted X-rays hit the photographic film, they leave a pattern of spots. If the atoms in the crystal are arranged in a repeating, orderly pattern, the spots on the film will form a very specific geometric pattern.
Okay, so it's not random.
Not at all. By applying incredibly complex mathematics, specifically Fourier transforms to the position and brightness of those spots, you can calculate the three-dimensional arrangement of the atoms.
And we should remember, doing that math in the 1940s and 50s meant doing it by hand. Like, no computers, just you, a slide rule, and endless columns of numbers.
Weeks and months of manual calculations for a single structure. And this brings us to what Franklin was doing in Paris after she left Cambridge.
Right, because she went to France.
Yeah, and she wasn't working on simple, perfect crystals like salt or diamonds. She spent 4 years at the State Chemical Laboratory in Paris, focusing on the X-ray diffraction analysis of coal and carbon structures.
Wait, coal isn't a crystal, is it? It's just a messy lump of carbon.
Which is exactly why her work was so groundbreaking. I mean, coal is mostly amorphous. It doesn't have a neat, repeating, orderly structure. It is chaotic.
Wow.
To get meaningful diffraction data out of a lump of coal using X-rays requires a level of experimental precision, patience, and technical mastery that very few scientists possessed at the time. She had to painstakingly prepare the samples, evacuate all the air from the chambers, and interpret incredibly faint, blurry bands on the film rather than nice, sharp spots.
She was basically doing crystallography on hard mode.
She was defining a new discipline. She discovered the fundamental difference between carbons that turn into graphite when heated and those that do not.
Oh.
Yeah, and her work in Paris was so rigorous, it was actually used to design better, safer gas masks during and after the Second World War by understanding the porosity of carbon.
That's incredible.
So she wasn't an assistant. By the time she left Paris to join King's College London in 1951, she was an internationally recognized authority in her field. She was brought in specifically to apply these exact rigorous techniques to biological molecules, namely DNA.
You know, her scientific temperament seems perfectly matched to that level of difficulty. She strikes me as someone who operates like a master architect.
I mean,
Like, she absolutely refuses to show anyone the blueprints for a skyscraper until every single load-bearing wall, every stress point, every material tolerance has been mathematically proven on paper.
Yeah.
She will not build the building until she knows for a fact it won't fall down.
that is a perfect analogy. She fiercely resisted premature conclusions. In her view, the empirical data had to lead to the model, not the other way around. You do not build a theoretical model until the data fully, irrefutably supports it.
But doesn't that careful, methodical temperament actually put her at a massive disadvantage? Especially in a scientific community that loves a fast, flashy breakthrough.
It can, yeah.
I mean, if you are meticulously checking your math while the guys down the road are throwing spaghetti at the wall to see what sticks, aren't you going to get beaten to the punch?
Well, her rigor was her greatest strength as an experimentalist.
Yeah.
But you are identifying the exact vulnerability that defined her time in London.
Right.
If we connect this to the bigger picture, she was entering a race against faster-moving rivals like Watson and Crick. They were perfectly willing to build speculative physical models early in the process.
Even if they were wrong.
Exactly. They were willing to be wrong, to guess, to iterate quickly based on partial data. Franklin wanted to be definitively, undeniably right. And in a highly competitive environment, that caution can easily be weaponized against you.
Which brings us to the actual environment she was walking into at King's College. Because we are taking this powerhouse of an experimentalist, pulling her out of a collaborative environment in Paris, and dropping her into a laboratory setup that feels, honestly, perfectly engineered to explode.
It really was. The setup at King's College was a masterclass in administrative dysfunction.
Yeah.
The laboratory was directed by a physicist named J.T. Randall. Now, Randall had secured funding to study the structure of DNA, and he recruited Franklin specifically to upgrade their X-ray crystallography unit.
Okay.
Before she arrived, he wrote her a letter explicitly stating that the DNA X-ray work would be her project and that she would have a graduate student named Raymond Gosling assigned to assist her.
Well, I mean, that seems clear enough on paper.
On paper, yes.
Right.
But the catastrophic failure of management happened because Randall failed to communicate this arrangement to the person who was already at King's working on DNA, Maurice Wilkins.
Ah. The man with the filing cabinet from our opening scene?
The very same. Wilkins was a very capable physicist who had actually worked on the Manhattan Project. He had initiated a lot of the early promising DNA X-ray work at King's.
Oh, I didn't realize that.
Yeah. But he was away on holiday when Randall wrote that definitive letter to Franklin. So when Wilkins returned, he found Franklin installing her own equipment, directing Gosling, and taking complete ownership of the project.
Oh, no.
And Wilkins, having never been told otherwise, just assumed she was brought in to be his highly skilled assistant.
Imagine being hired to be the head chef of a prestigious restaurant. You walk in on your first day, ready to design the menu and run the kitchen, but the owner totally forgot to tell the current head chef that you were coming to take over his station.
Right?
You weren't just fighting over what dishes to cook, you were fighting over who even has the right to stand at the stove.
That's exactly it. The resulting friction was inevitable. It was fundamentally a failure of leadership, but history has too often portrayed it as merely a personality clash, leaning on these tropes that Franklin was simply difficult to work with.
Right, which is so unfair.
It is. The reality is that two highly ambitious, capable scientists were structurally set up to fail. They were thrown into a small space and expected to collaborate while operating under entirely contradictory assumptions about who was actually in charge.
And that structural failure breeds an incredibly toxic, isolated environment. If you feel like your territory is constantly being threatened by the person sitting across the hall, you aren't going to sit down over coffee and collaboratively hash out a problem. You are going to hoard your findings.
Absolutely. The lack of a defined hierarchy meant that Franklin retreated into her own rigorous, independent work, and Wilkins felt sidelined and deeply frustrated. They eventually stopped speaking to each other almost entirely, communicating largely through formal, terse notes.
Wow. Just passing notes.
Yeah. And when the front door of collaboration is nailed shut by bad management, information tends to find a back door. This environment of isolation set the perfect stage for the clandestine sharing that would follow.
But despite all of this friction, despite the toxic environment and the lack of communication, Franklin's unparalleled skill still yields one of the most important artifacts in scientific history. In May 1952, she captures Photo 51.
And to understand the staggering technical achievement of Photo 51, we really have to look at the physical reality of DNA. It is a biological molecule. It is not a hard, stable crystal. It degrades.
Okay.
More importantly, Franklin discovered early on that DNA fibers actually exist in two different forms depending on how wet or dry they are.
Wait, really? So the molecule literally changes shape based on the humidity in the room?
Yes. Exactly. She discovered that when the DNA fiber is relatively dry, it forms a short, stubby, highly crystalline structure she called the A form. But when she exposed the fiber to high humidity, it stretched out. It elongated into a paracrystalline structure she called the B form.
Why does that matter so much?
Because before Franklin arrived, researchers were inadvertently taking X-ray pictures of DNA that were a mixture of both forms at the exact same time. The resulting images were just a blurry, uninterpretable mess.
Ah, that makes sense.
It was Franklin's brilliance as a physical chemist to realize she had to physically separate the two forms by rigorously controlling the humidity during the exposure.
But how is she controlling the humidity around a microscopic fiber in a drafty London basement in 1952? I mean, she doesn't have digital climate control sensors?
No. She designed a customized micro-camera setup. She would draw the DNA out into a fiber no thicker than a single strand of spider silk.
Wow.
She placed it inside a sealed chamber. To control the humidity, she bubbled hydrogen gas through specific salt solutions because different saturated salt solutions maintain very precise, known humidity levels in closed spaces.
That is so clever.
Isn't it? She maintained the humidity at exactly 92% to keep the DNA locked in the extended B form.
And then she fires the X-ray beam at it.
But the beam has to be incredibly fine, and because the sample is so small and delicate, a single exposure took an exceptionally long time, sometimes up to 100 hours.
100 hours for one photograph. If the temperature in the room fluctuates, or if the building vibrates from a passing truck, the image blurs.
Exactly. If the alignment was off by a fraction of a millimeter, the image would be totally useless. But in May 1952, working alongside Raymond Gosling, she developed a piece of film that had been exposed for 62 hours. The result was Photo 51.
Unbelievable.
Historians of science regard it as one of the clearest and most important X-ray images of a biological molecule ever taken up to that time.
And here's where it gets really interesting. She develops this film, and she looks at the pattern. It's a very distinct, heavy black X shape made up of dark dashes radiating out from the center of the film. And because she understands the math of how X-rays bounce off atoms, she knows exactly what she is looking at.
She knew immediately. In X-ray diffraction, a repeating zigzag shape in the physical molecule, which is basically what a helix is when viewed from the side, will always cast a cross-shaped pattern on the diffraction film.
Okay.
The fact that photo 51 showed such a stark, unmistakable X meant that the B form of DNA was undeniably a helix.
She knew it was helix. I want to try and put myself in her shoes. Standing in a darkroom in 1952, holding the physical proof of the secret of biological inheritance in your hands. She is methodically, quietly proving it to herself with absolute mathematical certainty.
And she deduced much more than just the general helical shape, too.
Way.
By measuring the angle of the X on the film, she could calculate the pitch of the helix, like how steep the spiral staircase was.
Right.
It was exactly 34 angstroms per full turn. By measuring the distance between the dark spots along the arms of the X, she knew the distance between the repeating units, the steps on the staircase, was 3.4 angstroms, meaning there were exactly 10 steps for every full turn of the helix.
She is basically reading the exact physical dimensions of the molecule straight off a shadow cast on a piece of film.
She was. And she also noticed something even more profound. If you look at the dashes that make up the arms of the X in photo 51, they are called layer lines. If you count out from the center, the fourth layer line is entirely missing. There's a blank space where a dark spot should be.
Wait, why would a spot be missing if the structure is uniform?
Well, in the mathematics of helical diffraction, a missing fourth layer line specifically occurs when two helices are intertwined, and they physically cross over one another at that exact point, canceling out the X-ray reflection.
Oh, wow.
Yeah, that missing spot told her that DNA wasn't just a single helix. It was a double helix. And furthermore, based on the water content required to maintain the structure, she had deduced that the bulky phosphate backbone of the molecule had to be on the outside of the helix, not the inside, facing the water.
So she had the helix, she had the dimensions, she had the phosphate on the outside, and she knew it was a double strand. She had almost the entire puzzle. Why didn't she immediately publish a paper screaming, "I found it"?
Because of that architect mentality we discussed earlier. She was not a model builder by nature, she was a rigorous empiricist. While the B form, the wet form, clearly showed a helix, her data on the A form, the dry form, was much more complex and stubbornly ambiguous.
So she wanted to be sure.
Exactly. She wanted to comprehensively solve the mathematical functions for the A form to definitively prove that both forms were helical before she published a sweeping structural claim. She was doing the grinding, unglamorous mathematical work required for absolute proof.
Which brings us back to that drafty office in January 1953, the tension in the lab, the brilliance of that photograph, and the ticking clock of rival researchers. It all collides in a specific, documented moment of unauthorized sharing.
Right. This is the moment Maurice Wilkins slides open the drawer and shows photo 51 to James Watson. Now, Watson was visiting from the Cavendish Laboratory at Cambridge, where he and Francis Crick were desperately trying to build a physical model of DNA.
And they were struggling, right?
They were completely stuck. They didn't know if the backbone was on the inside or the outside. They didn't know the exact pitch of the helix, and they weren't even entirely sure if it was a double or triple helix at that point.
But then Watson sees the photo. And because he is also a brilliant scientist, he knows exactly what that X shape means.
In his own memoir, The Double Helix, Watson describes his reaction vividly. He wrote, "The instant I saw the picture, my mouth fell open and my pulse began to race. The pattern was unbelievably simpler than those obtained previously. The photograph gave him the visual confirmation he needed. It proved the helix."
It is just an incredible violation of trust. But I've always wondered, is a single photograph really enough to build the entire atomic model? Could they have gone back to Cambridge and built the double helix just from looking at that one picture in a drawer?
No. And this is a vital part of the historical record that often gets completely overshadowed by the drama of photo 51. There was actually a dual nature to this data leak. The photograph was the spark, but the heavy lifting, the hard mathematical parameters required to actually build the physical model came from a second, completely separate, unauthorized transfer of Franklin's data.
Wait, what was the second lick?
It involved a researcher at Cambridge named Max Perutz. Perutz was a highly respected crystallographer, and he served on a committee for the Medical Research Council, or MRC. That was the government body funding both the Cambridge lab and the King's College Lab.
Okay, the funders.
Right. In late 1952, as part of his official committee duties, Peretz was given a copy of an unpublished internal research report summarizing the progress at King's.
Like an administrative progress report.
Yes, and Rosalind Franklin had meticulously prepared a section of this report. She summarized her exact findings on DNA. She included the precise measurements she had calculated from her photographs, like the 34 angstrom pitch, the 3.4 angstrom distance between bases.
All the hard numbers.
All of it. Most importantly, she included a piece of crystallographic data regarding the crystal symmetry of the DNA molecule. She identified it as belonging to a specific space group called monoclinic C2.
Monoclinic C2, what does that actually mean in plain English, and why is it the smoking gun?
Well, in crystallography, the space group tells you how the molecules are packed together in the crystal. The C2 space group possesses what is called two-fold rotational symmetry.
Meaning if you turn it upside down, it looks exactly the same.
Exactly. Imagine taking the DNA double helix and flipping it 180 degrees. If it looks identical, that mathematically proves that the two strands of the helix are running in opposite directions. One strand runs up, the other strand runs down. They're anti-parallel.
And Watson and Crick didn't know that.
They had no idea. They had been trying to build models where the two strands ran in the same direction, and the atoms simply wouldn't fit together properly without violating the laws of chemistry.
Wow.
So when Max Peretz took this unpublished internal MRC report and handed it to Watson and Crick, Francis Crick, who was a master of crystallographic theory, read the words monoclinic C2. He instantly realized what it meant. The strands had to be anti-parallel.
Wait, Peretz just handed them the report? Did Franklin know her internal progress update was being passed to the rival team working on the exact same problem?
She did not. She had no idea her quantitative parameters were being used to calibrate their physical models at Cambridge.
Put this in the context of the 1950s scientific community, though. Was this considered outright theft at the time? Or was there a blurry line of, like, collegial sharing among government-funded labs that just looks terrible to us in hindsight?
What's fascinating here is that historians and scientists genuinely debate the exact cultural norms of the era. Max Peretz later defended his actions by arguing that the report was not stamped confidential and that it was the very purpose of the MRC committee to foster collaboration between its funded labs.
Sure, I can see that defense.
And defenders of Watson point out that the data in the report wasn't an explicit secret. However, the historical consensus, looking at the totality of the event, is clear and undisputed. The sharing was highly irregular and absolutely unauthorized by the scientists who actually did the work. It breached the standard etiquette that unpublished data belongs to the researcher until they are ready to present it. Whether one chooses to call it a systemic breach of etiquette or intellectual theft, the undisputed fact remains. Her data was transferred without her consent, and it materially provided the exact parameters Watson and Crick needed to solve the structure.
So armed with the visual proof of the helix from photo 51 and the exact mathematical dimensions and symmetry from the Peretz report, the Cambridge team goes into an absolute sprint.
The timeline compresses dramatically. Having the parameters meant they didn't have to guess anymore. They knew the backbone was on the outside. They knew the dimensions. They knew the strands were anti-parallel. All that was left was figuring out how the bases, the A, T, C, and G molecules on the inside paired up to hold the two strands together.
And Watson cracks that part right, the base pairing.
Yes, he does. Watson, playing with cardboard cutouts of the bases on his desk, realized that adenine pairs perfectly with thymine, and cytosine pairs perfectly with guanine. They fit together like rungs on a ladder, keeping the width of the helix perfectly uniform.
It's so elegant.
It was a brilliant, elegant realization. Within weeks of seeing Franklin's data, Watson and Crick complete their double helix model.
And they race to publish. In April 1953, they published their findings in the journal Nature, which is essentially the holy grail of scientific publishing.
Right, and the publication of the discovery is where the legacy of this event is permanently cemented, and it requires careful examination. In that issue of Nature on April 25, 1953, there wasn't just one paper on DNA. There were 3 linked papers published back-to-back.
3 papers.
The first paper was Watson and Crick's famous one-page theoretical model describing the double helix. The second paper was by Maurice Wilkins, presenting his general X-ray data. And the third paper was by Rosalind Franklin and Raymond Gosling, presenting their detailed, hard experimental data, including photo 51.
Wait, if they were all published together, doesn't that mean she got credit at the time? She's in the exact same journal on the exact same day.
It is entirely about the framing and the sequencing. The editorial choices made at the time framed Franklin's and Wilkins's papers not as co-equal independent discoveries, but merely as supporting evidence for Watson and Crick's brilliant theoretical leap.
Because Watson and Crick's paper was placed first.
Yes. By placing the theoretical model first as the main event, Franklin's meticulous, painfully gathered foundational data was relegated to the status of a backup confirmation. In their paper, Watson and Crick included a notoriously vague acknowledgement.
What did they say?
They wrote, "We have also been stimulated by a knowledge of the general nature of the unpublished experimental results and ideas of Dr. M.H.F. Wilkins, Dr. R.E. Franklin, and their co-workers at King's College, London."
Stimulated by a knowledge of the general nature. That is an incredibly mild way of saying, we looked at your photograph and used the exact dimensions from your unpublished report to build this.
It created an illusion for the scientific community and the public reading that journal. It made it look as though Watson and Crick came up with this brilliant theoretical model, largely on their own, and then, fortuitously, the experimentalists in London happened to publish data in the same issue that confirmed their genius.
Wow.
In the eyes of history, Franklin was positioned as the assistant, providing the confirmation, rather than the pioneer providing the foundation.
So, what does this all mean? Her own data was used to beat her to a punch she was already in the middle of throwing. She had the blueprints, she was doing the final math, and they took the blueprints, built the roof, and got all the credit for the house.
I do need to state a necessary historical caveat here, because the history of science is rarely black and white. Historians actively debate how absolutely essential Franklin's specific data was to Watson and Crick's final model.
Really? Even knowing about the photograph and the space group data from the report?
Yes. The debate centers on inevitability. Some historians argue that given Watson and Crick's theoretical brilliance, their focus on model building, and other chemical data available at the time, they would have eventually reached the same conclusion on their own, perhaps months or a year later.
Okay, but still.
Others argue vehemently that they were completely stuck and would not have realized the anti-parallel nature of the strands without Franklin's C2 space group calculation.
But even if we acknowledge that debate, even if we entertain the idea that Watson and Crick might have figured it out eventually, it doesn't erase the reality of what actually happened.
Yeah.
The unauthorized transfer of the data is a documented historical fact. They used her work without her knowledge to get there faster.
Absolutely. The debate over inevitability does not negate the breach of scientific ethics. The unauthorized transfer is the undisputed core of the controversy.
It is vital, though, that we do not reduce Rosalind Franklin to this single narrative of victimhood. She is not just a tragic figure in the DNA story. She did not stop being a brilliant scientist just because these guys looked at her homework.
Far from it. Shortly before the DNA papers were published, Franklin had already decided to leave King's College. The working environment with Randall and Wilkins had become completely untenable. So she transferred to Birkbeck College under the direction of the crystallographer J.D. Bernal.
What was the environment like at Birkbeck?
Physically, it was a massive downgrade. She was placed in a small, cramped laboratory in a heavily bombed-out Georgian building. The roof literally leaked.
Oh, geez.
But culturally, it was a revelation. Bernal respected her immensely. She was given her own research group, and she turned her formidable intellect toward a new, arguably even more complex challenge: the molecular structure of viruses.
Viruses are massively larger and more complicated than a single DNA molecule, right?
Exponentially more complex. She focused on the tobacco mosaic virus, or TMV. TMV is a massive rod-shaped structure made of thousands of protein molecules surrounding a core of genetic material RNA. Before Franklin, the structure was poorly understood.
How does she map something that large using the same X-ray techniques?
She applied the exact same rigorous, meticulous methodology she had honed in Paris and at King's. She took the highest resolution X-ray diffraction images of the virus ever achieved, and her analysis yielded a profound discovery.
Which was?
At the time, the leading theory, championed once again by James Watson, who was also studying TMV, was that the virus's genetic material, the RNA, sat on the outside of the protein structure. Franklin's precise crystallographic math proved definitively that Watson was wrong.
Huh.
She proved that the RNA was a single, continuous strand embedded deep inside the hollow protein tube, winding through it like a wick inside a candle.
She proved him wrong with math again.
She did. And she didn't stop there. She did foundational structural work on the poliovirus, which is the first animal virus she had ever tackled. Polio was a crystalline sphere, incredibly difficult to analyze. Her work on viruses was so highly regarded that she was asked to construct large-scale models of viruses for the 1958 World's Fair in Brussels. She was leading a heavily funded research team, securing international grants, and making massive foundational strides in structural virology.
Think about how easy it is to reduce brilliant people, and especially brilliant women in history, to the one time they were wronged. We turn them into a martyr for a single cause, we memorialize the theft, but when we do that, we risk ignoring the entirety of their towering, independent achievements. Her post-DNA work proves definitively that her genius wasn't a fluke. She was a titan of structural biology entirely on her own terms.
Her virology work entirely validates her method. It proved that empirical rigor, while perhaps slower in the short term, produces unassailable foundational truths. She laid the groundwork for modern structural virology.
Which makes the final chapter of this story so incredibly difficult to process, because it doesn't end with a neat resolution. There is no later vindication during her lifetime where the scientific establishment apologizes and recognizes her foundational role in DNA. It ends with a sudden, devastating tragedy that permanently foreclosed any chance of a historical reckoning while her peers were still alive.
In the fall of 1956, while she was at the peak of her virology research, Rosalind Franklin was diagnosed with ovarian cancer.
She was in her mid-30s, right?
She was 36 years old.
Oh.
And true to her nature, she did not stop working. She underwent multiple surgeries and experimental treatments, but she continued to direct her research team. She continued to publish papers. She continued to seek funding for the polio research right up until the very end. She died on April 16, 1958. She was only 37 years old.
37. It is a heartbreaking loss to science. Is it true that her cancer might have been caused by her work? I mean, she was working with high-energy X-ray radiation for hours on end, day after day, for over a decade.
There is a layer of speculation there that we must address carefully. Some commentators have indeed theorized that her early cancer was linked to her extensive occupational exposure to X-rays, particularly during her early years in Paris and London, when shielding protocols were far less stringent than they are today.
Right, that makes sense.
However, cancer is a deeply complex disease, and she also had a family history of cancer. We cannot say with absolute scientific certainty that the X-rays caused her illness, but given the nature of her work, it remains a haunting possibility.
Her death sets the stage for the final, unanswerable question of this entire saga. Because 4 years after she dies in 1962, the ultimate prize in science is handed out for the very discovery she laid the foundation for.
In 1962, the Nobel Prize in Physiology or Medicine was awarded jointly to James Watson, Francis Crick, and Maurice Wilkins for their discoveries concerning the molecular structure of nucleic acids, the structure of DNA.
And Rosalind Franklin is nowhere on that list.
She is not. And this raises an important question of recognition, but it is one we have to look at strictly through the lens of the Nobel Foundation's own statutes. Under their explicit rules, the Nobel Prize cannot be awarded posthumously. You must be alive to be nominated and to receive it.
So even if the committee had wanted to give it to her, even if Watson and Crick had stood up and demanded Rosalind Franklin be recognized alongside them, the rules dictated that she was entirely ineligible.
Precisely.
And that is such an unsettling reality. We all want the neat narrative where we can definitively point to a villain with a gavel and say, the committee robbed her. We want a clear injustice we can rage against. But the mechanics of the rules meant she was disqualified by the tragedy of her own biology before the historical reckoning could occur. It leaves us with this permanent echoing what if.
It is the great unanswerable question of 20th century biology. We know her data was essential. We know it was transferred without her permission. We know she was an experimental scientist of the very highest caliber who was closing in on the answer herself. What we will never know is how the institutions of science would have ultimately judged her contribution if she had lived to stand on that stage in Stockholm alongside them.
Let's take a breath and recap the journey we've just been on. We started with a remarkably rigorous scientist, a master of physical chemistry who thrived on mathematical certainty. We watched her get dropped into a deeply toxic, mismanaged laboratory environment at King's College. We explored the staggering experimental brilliance required to control humidity and radiation to capture Photo 51. We saw that visual data and the crucial mathematical parameters of the space group transferred in secret to rival scientists,
which provided the foundation for a theoretical model that fundamentally changed the course of modern biology and genetics.
We tracked her independent rise as a titan of virology, mapping the unseen structures of disease. And we confronted a brilliant life cut tragically short at 27, leaving a legacy that is still being debated, reconstructed, and honored today. I want to leave you with a final thought to mull over on your own, and it goes back to how we consume information and how history is written. Think about the sheer power of the table of contents in a magazine or a scientific journal. In April 1953, Watson and Crick's theoretical paper was placed first. Rosalind Franklin's paper, containing the actual physical proof, was placed behind it, framed by editorial choice as mere supporting data. If the editors of Nature had simply arranged those pages differently, if they had published her hard experimental data first, establishing the undeniable physical reality of the molecule through her grueling labor, and placed their theoretical model second, framing it as an interpretation of her foundation, how would the history of biology have been written in our minds? Does history actually remember who did the fundamental work, or does it only remember who got to write the headline? Thank you for taking this deep dive with us. Keep questioning the headlines, and we'll see you next time.